It's Dr. L, and we are going to continue learning about phospholipids and membranes, focusing really on the basic chemistry. In the previous video, you learned about phospholipids and the term amphipathic. Now let's take that knowledge back to the membrane in a cell. So this is a really nice cartoon I found of a cell membrane. The phospholipid molecules are shown in their bilayer conformation. It's a lipid or phospholipid bilayer, and in gold we have like the little phosphate heads, and we have the two little kind of grayish brown or dark beige fatty acid tails in each of them. And then we have a big teal colored protein that is a transport protein. It's a transport protein for water. The water molecules are shown and they're labeled. They're little red oxygen and the two gray hydrogens. And this particular protein is a water transport protein. It could be the protein called aquaporin, aqua, A-Q-U-A, porin, P-O-R-I-N, which is a protein found in many different cellular membranes that's specific to facilitate the transport of larger quantities of water across the membrane. It's only good for water. That's what it's there for. And then we can see that there are water molecules on the outside of the cell and there are water molecules on the inside of the cell. So the cytoplasm is water-based, and outside of the cell, all cells need to have at least a thin layer of water in order to be alive. Water molecules can passively diffuse across membranes, but they do it slowly because of their partial charge, which is what drives the need for proteins like aquaporin that lets more water molecules move across a little bit faster. Here, what I want you to think about is the fact that all of those phosphate heads on each individual phospholipid is hydrophilic. It's got some charge associated with it, in this case a full negative charge, but also some partial charge due to the polar covalent bonds within the phosphate groups. That means that those phosphate heads are hydrogen bonding with all of these water molecules shown here. They're going to be forming and breaking, forming and breaking. And it is that chemical bonding with these water molecules that keeps the phosphate head pointing towards the water, therefore pushing the fatty acid tails away from the water. Same thing's happening in the lower section of the phospholipid bilayer. The phosphate heads are hydrogen bonding with the water molecules inside the cytoplasm, and those bonds are forming and breaking, but there are enough of them that they keep the phosphate heads outwards, or inwards in this case, pointing towards the water, which pushes the fatty acid tails towards the inside. We are also going to have another type of chemical bonding between the fatty acid tails, which are very hydrophobic, lots of carbon-carbon, lots of carbon-hydrogen bonds. Those are non-polar covalent bonds. We're going to have what are called hydrophobic interactions happening here, and that's helping to stabilize the membrane as well. Okay, so hydrogen bonding happening between water molecules themselves, between water molecules and the phosphate heads, on the outside of the cell membrane and on the inside, and then hydrophobic interactions happening between the fatty acid tails within the lipid bilayer kind of sandwich structure. So these lipid bilayers form spontaneously whenever you put phospholipids into water or a water-based solution. In cells, we see formation of these bilayers as shown on the bottom of this figure. But if you were to just drop some like phospholipid solution into like a small beaker of water, if it was a small amount, what we would see is the formation of like these little tiny structures called liposomes, or even these littler structures called micelles, where just the little fatty acid tails interact, forming just a single layer of phospholipids. I point out liposomes and micelles because they play important roles in our digestive tract in the absorption of certain vitamins and minerals. You can learn more about that in a nutritional biochemistry class if you're interested, or look it up. I also point out the fact that we take advantage of these structures in order to deliver some drugs into human and other animal systems. So this is used pharmaceutically, this chemistry. okay, not just like naturally and biologically. Lipid bilayers make up about 50%, maybe a bit more, of the mass of any cell membrane. So the other stuff that's in cell membranes is largely protein-based. Cytoplasmic membranes bound the cells, so all cells have them. They are selectively permeable, meaning they control what goes in and out of the cell. And they're also important in metabolism and therefore maintaining cellular homeostasis. Those are all important functions of the cytoplasmic membrane, and all cells have cell or cytoplasmic membranes. A couple of additional points about the cytoplasmic membrane is that those fatty acid tails, so this is a drawing, it's a space-filling drawing of a phospholipid. So it looks a little different from the previous pictures, but it's the same basics. There is variability in the length and the chemistry of the fatty acid tails in different species of organisms. And there can also be variability in what additional chemistry is attached to the phosphate head between different species of organisms. And that's like more detail than we need to get into here, but I'm pointing it out for future learning. Another thing I'll point out is that the phospholipid membranes of bacteria versus archaea and also versus eukaryotes differ. So what we see is bacteria and eukaryotes build their phospholipids like what we see in this bottom figure. We'll tend to see ester linkages connecting fatty acid tails to a glycerol backbone, which is then attached to a phosphate head, and then sometimes you can have some additional chemistry added to that. They don't always have unbranched fatty acids like this drawing is suggested. They could be branched, they could be unbranched, but the key here is they have ester linkages. In contrast, all of the archaea, they don't have fatty acids as their tails. They have these structures that are really similar but a little different called isoprene chains. And they connect their isoprene chains, which are also hydrophobic, just like fatty acids. They connect those isoprene chains to a glycerol backbone using a different chemical linkage called an ether linkage. And then they also use a phosphate head. So that's, again, a lot of picky detail for this level of video and instruction, but I'm pointing it out in case you're curious. And I'll point out one more thing, which is in the microbial world, there are microbes that love to live where it's really, really cold, and there are microbes that love to live where it's really, really hot. And they're not just surviving at those temperatures, they're actually multiplying. So neither of these groups do well at the temperature that our bodies are at, which is 37 degrees Celsius. Cold-loving microbes are called psychrophiles, and hot-loving microbes are called thermophiles. And we tend to see differences in their phospholipids, specifically the fatty acid tails that they use. So some of them tend to have more saturated fatty acids, which pack together tightly, and others have unsaturated fatty acids, which stay like a little bit looser and use more space. Organisms that live where it's really, really cold tend to use more of these unsaturated fatty acids, and organisms that live where it's really, really hot tend to use more of the saturated fatty acids. And that is because the saturated fatty acids give these phospholipids, they allow those phospholipids to form lipid bilayers that are more stable at high temperatures because they're so packed together. And so they don't like melt apart at high temperatures. Whereas where it's really cold, the risk is that the membrane will freeze, and to prevent freezing, you want to have phospholipids that are moving around a lot more. So you build your phospholipids out of fatty acid tails that are like contain more of these unsaturated C double bond C bonds. Again, more detail than we really need for this particular level of video, but just some interesting extra biology you might be curious about. And that pretty much sums up what I'm going to include here about phospholipids and about membrane structure. But I'm going to point out that all of this chemistry that I've been covering in these last 3 video lectures will help you understand the transport of molecules in and out of cells, which is necessary for cells to do biochemistry.